Okay, good morning, good afternoon, and welcome to Biomass Canada Cluster Project Update Webinar Series. My name is Mahmoud Abadian. I'm a research associate at the University of British Columbia working on biomass and bioenergy logistics and supply chain. And I work with Biomass and Bioenergy Research Group, as well as the Forest Product Biotechnology and Bioenergy Research Group at UBC, and I will be moderating today's webinar. I just want to mention that in the last 2 years, we had 2 webinar series. The first one, we had the updates of the different projects we have in Biomass Canada cluster. That was the first project updates webinar series. The second webinar series we had, we work with Agro Bioheat and Bioenergy Europe to invite speakers from US, from North America, and also from Europe. They talk about their experience of using agricultural biomass mainly for bioenergy production. We also had some webinars on biomass production. We had some of the discussion on policy development. So if you are interested to take a look at the slide and also watch the recording of the previous webinar, just please go to the webinar section of the Biomass Canada website, and you can find both the presentation slide and the recording. So this webinar series that we start today, we are going to focus again on project updates from Biomass Canada cluster. So we have 5 webinars. And in each webinar, we have two projects, and the project leaders are going to give the latest update about their project. And as before I actually introduce the speakers, I just want to mention, just give you an overview of the Biomass Canada cluster. I'm sure if you have attended the webinars before, you already have seen these slide bar. For those participants, they haven't heard about the Biomass Canada cluster. So this is a cluster that established in 2018 and is led by Biofuel Canada Network and has been financially supported from Agriculture and Agri-Food Canada's Agri-Science Program, and of course the contribution from industrial partners as well. So the focus of this cluster is just to increase the contribution of the Canadian agriculture into growing Canadian bioeconomy. And how we can do that, we are trying to de-risk and commoditize agricultural biomass and make it available feedstock for different low carbon products, including bioenergy, biofuels, and also other bio-based products. So that's, and I forgot to mention that actually we have 10 activities or projects. And again, in the next 5 webinars, we are going to talk about all of these activities and the progress they have made so far. So for today, we have update from 2 activities. Activities number 8, which is led by Dr. Donald Smith. He's a distinguished James McGill professor at the McGill University. And then, so that's going to be about half an hour presentation. And then we have about 15 minutes Q&A. And for the second presentation, we have Dr. Animesh Dutta from University of McGill, Guelph University, sorry about that. He's a professor at the University of Guelph and director of the Biorenewable Innovation Lab. Also, he has his colleague, Dr. Ranjan Pradhan. He's also a special graduate faculty and project manager from the same lab. They are going to give the latest update about the activity number 5. I forgot actually to mention that the activity 8 is focusing on the development of biological as low input, sustainable production practices for fuel and residue and food production. That's again the activity that Dr. Smith is leading, and Dr. Dutta is actually leading the activity number 5, which focuses on upcycling of agricultural waste to energy, chemicals, and materials. So before I hand it over to Dawn to start his presentation, just 2 quick housekeeping items. So this webinar is being recorded, and you can find a copy of recording and a slide after the webinar on Biomass Canada website. And also, if you have any Q&A question, please type it in the Q&A box, not in the chat box. And then after the presentation, we try to answer as many of these questions we can based on the time we have. So with that, I'd like to hand it over to Don, and we're looking forward to your presentation, Don. You are muted, Don.
Sorry, thank you very much. Yeah. Okay, so I'm just going to get this up here. Come on. There we go. So I'm just going to move the everybody's image off to one side here a little bit, and I'm going to see if I get my laser here. Yes, it looks like I can. Okay, so is it okay if I go ahead and start then?
Sounds good. Yep.
All right, I shall do it. I'm just going to. Oh, sorry. I'm just going to. Sorry. I'm just got a timer running here so I can tell where I am. Okay, here we go. So yeah, so I just start with the acknowledgements. We're funded by the Government of Canada under the Canadian Partnership Program. This is what the whole thing, the biomass cluster, runs on. And Biofuel Net Canada is the not-for-profit organization that holds the actual cluster, manages it, et cetera. In my case, there are three industrial partners here. AgroWorld, which is a startup, actually was started doing some work on flavonoids and was just starting in it. We talked to it about some of the work we're doing with microbes, and they folded that into what they were doing as well. Ulysses Biotech, and these are both agricultural companies. Okay, doesn't want to go up and down. What's it doing? There's always something, right? Okay, all right. The goal statement, yeah, and there are several of these because you'll see there are several threads that run through this. So first is for a microbial consortium already established. So this is one from the Ulysses company. Control environment work, evaluate its ability to improve production of biomass crops, crop residue, food material. We'll talk about that, field work as well. And we bioprospected to find plant growth promoting material, and so we'll talk about that. Phytomicrobiome, this is microbes associated with plants, bacteria in our case, and we assess their potential to become technologies for agriculture. Investigate mechanisms of action for the growth promoting bacteria involved, and we've done some of that. There's always more you can do, but we've done quite a bit. And then we tested on crops. So you'll see we got biomass crops, switchgrass and miscanthus, and we've got crop residue testing, generation of crop residues, corn and potato. And we've got, I think we also at some points, we've got a bit of control environment work with both canola and soybean here. So yeah, project overview. This is biomass crops could be grown on marginal lands in Canada. So this is greater than class 3, 4, 5, 6, 7. If you grow them there, they don't compete with food crops very much. We don't want to be competing with food crops. Not a good idea. And they just, once you get greater than 3, they're usually not growing food crops on that land. However, it does mean that the plants that are growing there are more likely to be stressed. These are marginal soils because the area is too dry, or the soils are too light, or too shallow, or something, right? So the plants are likely to be more stressed because they're on it. Perennial crops, these are once the stand is established, they can be harvested for anywhere from 15 to 30 years. And what can happen then is you can be generating fuel from the biomass, and you can also be increasing the carbon or organic matter content of the soil, assuming you're managing them well. So you're actually putting more organic matter in there than the wild stuff that was there before was. And I did a rough estimate. We could probably produce about 25 megatons or million tons of biomass this way, which is not trivial, each year. And then food crop residues. For that, we could take about a third of the residues each year. If you take more than a third, you start to deplete your soil carbon. So then you're transferring carbon from the soil into the atmosphere through a very convoluted route, and it's just not something you want to do. So you can take about a third a year, or maybe all of it every third year. But here there's about 48 megatons of biomass. So this is quite a lot of biomass altogether. PGPR, this is plant growth promoting rhizobacteria. These are the ones that are associated with the plants and can promote growth, as their name suggests, can help deal with stress. A lot of them do, and can increase yield as a result. So climate change resilience could be part of this, helping to deal with stress. And if it's increasing production of the plants, if it's increasing crop residues, food crop residues, and food of those crops, so it's not the standard food versus fuel conundrum. Instead, it's food and fuel. So this is just the opportunity, saying here, there's these yellow parts here on the map are the more marginal soils. We're the perennial grasses, purpose-grown crop biomass produced on these areas, and this could be an interesting use of those areas. So the approach, the problem, the opportunity here is the uncertainties about biomass feedstock supply were identified when we were running BiofuelNet as an NCE, and it was identified. The supply was identified as a major bottleneck. In 2014, there were a number of, I think there were 3 or 4 biomass plants built in the American Corn Belt based on corn and other crop residues. And all 3 of them, I think there were 3 there. I think there was one in Florida or something else, but all 3 of them went broke because ability to get reliable biomass access. So this is a major stumbling block. So we focused the biomass cluster on biomass production. So purpose-grown biomass and stress, though, is an issue. It's produced on marginal lands, as I said, so there's more stress more often. And increasingly frequent climate change conditions are going to lead to more stress more often that way, too. At the same time, many plant-associated microbes help plants deal with stress. This is becoming increasingly well demonstrated. So there's an opportunity there. And as I said already, you can take about 1/3 of the crop residues each year. But again, with even on conditions where you're not on marginal soils, you could be on really good soils, there's still going to be more climate change induced stress from time to time. So PGPR, again, could help with that, food and fuel. And this is just an example of what you can see, phytomicrobiomes and stress. So this, what we have here, if you look over here, for instance, this is Arabidopsis, not a crop plant at all. And we were growing it, say here, on Petri plates with signals, 2 different signals, but these are both signal compounds that we worked on. LCO was isolated by somebody else, but in the context of symbiosis establishment in nitrogen-fixing legumes, we discovered that it also helped plants deal with stress. It fired up stress mechanisms, stress response mechanisms in the plants. And Thyristin 17, we isolated the microbe that produces it. We identified it. We isolated the compound. We identified it. So, and what you see here, this is salt stress, and we often use salt stress just because it's the most easily applied stress. You can apply it uniformly and easily, quickly. So we often use it. Sometimes we use something else, but often we do this. So here it is. This is no salt stress, and this is salt stress, 150 millimole salt stress. That's reasonably stressful for Arabidopsis. And you can see it's reduced the plant growth to about 10% of here. And here we are, the same level of salt. We've added lipocyto-oligosaccharide, these LCOs. And you can see it's largely or totally restored the growth. This technology has already been commercialized. It went through a long, convoluted chain of events, and it's with Bayer now. And I think it's been going on about 100 million hectares of agricultural land each year. Until recently, that Bayer has made $1 billion off it. I don't know if that's true or not, but that's what they told me. And this one's Thyristin 17. It's a small peptide. This is actually a lipocyto-oligosaccharide. It's a complicated molecule. This is a small peptide, and they both work at really low concentrations. This one's all the way down to 10 to the minus 11th molar. It's kind of the low end of hormonal. So they're impressive in that sense. You don't need much. And here they are. This is here. It's water stress induced with polyethylene glycol. See, at the right concentrations, you can restore most of the growth of the plant in the same way as here. All right, so yeah, so they're from microbes that are associated with plants, part of the phytomicrobiome, that population of microbes that are always with the plants, so the plant microbiome, and they help in a range of ways, nutrient mobilization, hormone production, disease control, and signals. And the signals often trigger stress responses in the plant, so the plant can deal better with stress. And we've been doing, as I said, quite a bit of work in that area. So one of the things we did, we went looking for some new PGPR, and I became aware quite a few years ago of there's a legume that lives here in this area. It's called Amphicarpi bractida, and it's the closest relative of soybean in North America, or at least this part of North America. If you look at the leaves here, if you work with soybean, you go, oh yeah, those are soybean leaves, or very close to it anyway. But it's a vine. So it's an interesting plant, and here you can see nodules on the roots of it. We collected not that many plants. We isolated microbes out of the nodules, and so we got the strains from there, from this indigenous legume. We focused on the 3 most promising ones, and you can see here. This is AB3. That's Amphicarpi bractida 3, which was I don't know what happened somewhere in the lab. Students renamed them all SL for Smith Lab 43. So I can see AB3 became SL 43. I don't know where the 4 came from. The 3 is obvious, AB3, but I don't know. Anyway, I'll ask them one day. But anyway, here they are. This is the control, and this is with the microbe, and this is without any salt stress. You can see it's not making a great deal of difference, et cetera, and the plants are doing well. But this is with salt stress, and you can see here, these plants are hurt badly, and here there's quite a degree of recovery. So this is our first observation of this kind of activity with this. So it seems to excrete a material into the medium because this is without the cells present. And what we do actually in this case is we'll grow them in a broth medium, a liquid medium. Then once they achieve a good population density in the medium, we'll centrifuge down the cells and then we'll filter sterilize it. And we use that material then has acquired the ability to stimulate growth because presumably something's been excreted into the medium. So this is, say, their exudate is enhancing stress tolerance in this. Okay. And here we are, progress to date. This is the new PG pair. This is the other two. The other two work well. The other two were AB2 and AB8, or SL 42 and 48. And they you can see here not too much going on at optimal growth conditions, but under salt stress, clear benefits from them. And so they helped quite a bit in this. And intellectual property was filed on these and the other one. And they've all been, I don't know if they're fully patented now or close to. I think I'm not sure what it is exactly, but anyway, the 2 most promising strains for inoculation, at least, the 42 is a rhizobium, and the 48 is a hydronophagia. And they've been investigated for soybean in promotion of salinity tolerance. And here we are. We inoculated with Bradyrhizobium japonicum, the nitrogen fixing bacteria associated with soybean, plus in this case SL42. Resulted in higher shoot biomass in the control, and this is sort of 18% at the vegetative stage, 16%. You see it's gradually going down as the plant seems to get adjusted to things. But even by the time you get to the end, there was still a 4.3% increase in total seed weight. So it's nice. With the other 48 or 48 plus, in grain yield is 7.4 and 8.1% at the end. So these all are promising, and are both look promising in that sense. So it suggests that inoculation with bacteria from an indigenous legume can induce stress tolerance, improve growth, yield, and support sustainability, and encourage broader ecological adaptability of soybean. It could grow under more stressful conditions. If I'm talking too fast, you guys should just warn me because I'm prone to that. And they always say, the more interested I am about something, the faster I talk, and the more caffeine I drink, the faster I talk, and I'm drinking this caffeine from time to time. All right, and this is just it's one of the graphs, just so you get a sense of what the data looks like. This is unstressed, and you can see the control and the effects of the two bacterial strains, and this is stress. It's in both cases actually. It's increasing fairly well. So we also did some proteomics on this. The soybean plants, again, the same 2 strains. Bacteria triggered multiple signaling pathways that regulated growth. A lot of this was energy metabolism, and also stress tolerance mechanisms. So this resulted in, it's a beneficial plant microbe interaction and enhanced plant growth. And when the plants were co-inoculated with these guys, the 2 strains we talked about, plus the Bradyrhizobium japonicum, the effects were greater than when they weren't co-inoculated, which suggests that these 2 guys may be helping the Bradyrhizobium japonicum in some ways. And that's an interesting situation, because if they can help nitrogen fixation, that's another good way to reduce greenhouse gas emissions. Okay, so under stressful conditions, they could effects boost crop productivity. So this is all looking quite promising. I must say, when I started putting these slides together and going through them, I realized over the last 4 years, we've come quite a distance with some of this stuff. So it's encouraging. The last strain was Devosia. The previous 2 types, we know the genera. We did 16S. The 16S suggested species for them, but we have to confirm that by doing total organismal sequence, DNA sequencing. But the Devosia looks like it's a new species. So we'll have to describe it. But anyway, this is the Devosia. It's a slow grower. It appears to be producing a growth stimulating substance, as I said. So we grow it in culture, broth culture, then remove the cells by centrifugation and filtration. But the resulting cell-free supernatant stimulates growth. And so that the cell-free supernatant, the CFS, and flavonoids, because the company, the AgriWorld, the partner, was also interested in flavonoids. So we applied them separately or together as seed treatments or as sprays, but this is a little bit on flavonoids. So these guys are involved in stress management, allelopathy. They can mitigate things like reactive action species problems. And they can act as plant to microbe signals, and again, in the establishment of the nitrogen fixing symbiosis. So the flavonoids we used was a citrus-derived set. There was a citrus extract of flavonoids was given to us by the industrial partner who have experience in this area. So this is kind of how this is. We did seed treatment, and we did foliar spray as well. So here are seed priming. So the seeds were soaked with solutions, flavonoid or the CFS for a specific time. Shorter for soybean because the seed coat, if you start soaking soybean seeds for a while, the seed coat degrades badly. So 3 hours is hours. Then we dried them back to their original weight. In every case, the treatments were compared with two controls. There was a prime control was soaked with water and then dried back to its original weight, and an unprimed control which was not soaked. And we had a range of treatments. It was all follow the same pattern. Here we have a set of flavonoid treatments. So you have a set of concentrations of flavonoids, and a dilution series, so to speak, and some salt levels. In each case, there's a zero control here for flavonoids, a zero for salt levels. Same here, this is for our cell-free supernatant. There's a set of dilutions and plus or minus salt. So there's a control in each case. Then this is the CFS plus the flavonoids, the two together, and again, the same sort of mixes. And the same thing here for soybean. Am I talking too fast? I'm okay or not? I worry.
Well, sounds good. That's great.
Good to know.
Thanks.
All right. So this is foliar spray. So in this case, the experiments were conducted this was in the greenhouse. Crops were sprayed only once, and they were sprayed. You got them what was roughly 1 mil per plant, and they were sprayed when they had enough leaf area. For canola, this was by the 4 to 5 leaf stage, and soybean by the 3rd trifoliate. And again, the same sets of combinations of treatments. So we had a range of flavonoids plus or minus stress. So, and if we look at this, is just some of the very beginnings of stuff we were doing with the flavonoids. This is the control with salt stress, but no flavonoid added. Here we are slightly more salt stress. But you can see here with the flavonoids added, they're germinating, and here they're not. So you start to see some benefits. We get more deeply into this with the seed germination work we carried out, which is more complicated. Seed and canola seed germination, as I said, or soybean and canola. The seed priming with flavonoids, potential biostimulant activity of the cell-free supernatant, this RCFS, right? Alone or in combination, so both of them, both together or each separately. And again, the presence of the salt stress. So there was a significant increase in canola and soybean seed germination caused by both these treatments, the flavonoids and the CFS, under both optimal and salt stress conditions, but generally the increases were greater under salinity stress. So that looks very interesting. Oh yeah, and the other thing was, yeah, well, okay, we'll come back to this. This is what the data can look like. It's interesting. This is optimal. This is no salt stress. This is no salt stress. And what you see here is that what you'll see eventually, you say this is in the presence of salt stress here and here, and this is soybean, and this is canola up top. And what you see is that these seeds are germinating in Petri plates, and eventually, even though there's salt, pretty much all germinate. It just takes them longer because they're stressed. So what you see is differences at the beginning. And you can see here, we've got these various treatments. You say, okay, yeah, there's a little benefit to having these microbial treatments and flavonoid treatments here. Here, actually, you can see that for soybean, these concentrations are too high. And it's interesting because as the concentrations become less and less, the effects are appearing, maybe, and it seems to be still going up. So maybe we should eventually try some even more dilute concentrations. So it's again an interesting thing how the small concentration these things work at. But when we go over to the stress situation, we see it, especially at the beginning, we get very large differences because of the additions of these materials. As I say, here in Petri plates, they'll all germinate eventually. Everybody that's going to germinate will. Under field conditions, it can be very different. If things germinate slowly, they may never make it above ground. And so having something like this that helps things germinate more quickly can make a big difference between having a very patchy stand and a very even stand. An even stand is what you want. If you get a patchy stand, you're just going to live with low yields at the end of the season. That's just how it is. So you want to have a nice even stand, and this is the kind of thing this kind of technology can get the plants going and out of the ground quickly and get you a nice even stand. Okay. So this is the we're seeing the flavonoids plus the Devotion, and well, this is with the spraying. CFS contain active compounds. Foliar spray determined growth stimulation effects in canola and soybean under optimal salt stress condition and salt stress conditions. This with the seed germination, both of the treatments affected the seed germination, and it was greater under the salt stress, and it's pretty straightforward. Much less so with the leaf applied material. So flavonoids under optimal conditions, soybean growth significantly affected, enhanced, increased shoot fresh weight, dry weight, leaf area. Here we are, 91, 99, 70. Those are nice big increases. However, soybean growth was unaffected by flavonoids when there was salt stress. You go, wow, so no effect with salt stress. That's not what you would have guessed. CFS, on the other hand, didn't have much effect when there wasn't salt stress, but when there was salt stress, it caused increases in here we are, growth, overall increasing shoot fresh weight, dry biomass production, and leaf area. 128, 163, and 194%. Those are very meaningful increases. So it's an interesting situation. And canola, on the other hand, just was much less responsive to the leaf sprays. I think we have some more work to do here, but it's a very interesting beginning, I must say. And this is just, again, so you can see the data. This is soybean, and with the CFS being added, and you can see nothing much happening in of stress, but in the presence of stress, here we are, control, and not much increase here, but nice solid increase here. So you're getting these kinds of things. So the right levels, you can have some, this is again the CFS, you can have some nice increases. This is a biomass field crop trial. So we're onto that part of it now. So trials planted in 2019, and we've done some other little plantings since to investigate specific things, but these ones we've been following. Each grass was treated with various microbes, either was the consortium or one of the new ones we were working on. Effects for established stands. The spray application enhancements occurred about half the time. So again, interacting with climate, and we'll see, we'll talk about corn and potato in a few minutes. Same thing. You'll see it doesn't always work. If the growing conditions are really good and there's not much stress, you don't see much effect. But if they're stressful, you do see effects. So as with other crops, probable interaction with environment stress right here. The levels of increase varied. They could be as low as a few percentage points and as high as 20%. But yeah, in the end, they can help make the crop more stress tolerant, especially, and you're seeing bigger effects when stress is present. And this could be very useful if they're being produced on marginal lands, as I said. This could be also applied to food crops. And actually, there's an interesting thing here. If you can do this, you can make the food crops more tolerant of stress, then you might be able to start producing it on some of the marginal land that's just barely marginal. So some of the marginal land might no longer be marginal because you can now produce successful food crops on it. So it's an interesting situation there. Okay, this is the last of these categories here at the end. This is a company, two companies that were working together, but this is Synagri and Ulysses, but they had a set of strains, a commercial consortium, and they were going to apply it with chemical fertilizer. So the chemical fertilizer became the carrier. But this is really worrying because you're basically adding on stuff that has a huge salt level. So we tested it and showed the strains actually survived. So we went ahead with it. And actually what we applied it at higher levels of inoculation, twice as high for the strains, because we were worried there'd be at least some cell death. But the recommended rate seemed to be the most affected in the end. So they seemed okay. Caused meaningfully and statistically significant increases for both potato and corn. And that's production of both the crop themselves and the residual biomass. The effects varied, suggesting an interaction with environmental conditions. And we'll just so this is corn and potato were grown. He said for a number of years. Samples are being processed now, and papers are being written at this point. But microbial strain, a group of 5 bacillus strains at various concentrations, 1 and 2x for the reasons I mentioned earlier. With the corn, we did it at 2 seeding dates, sort of recommended and late. We're trying to make sure we encountered stress at least sometimes. And we also had them on 3 soil types. We had them on heavy soil, we had them on a medium soil, and we had them on a light soil. And so we're just trying to ensure that we ran into stress somewhere. So the key findings, this is the first year on clay soil, clay loam. This is the intermediate soil, about 20%, and on sandy soil, about 13%. So both grain yield and starch content of the grain were increased by the microbial inoculant. So again, a lot of interesting things going on there. The relevance of this kind of thing, Research demonstrates the impact of a set of plant growth mycorrhizal bacteria, members of the phytomicrobiome, and the overall potential of the phytomicrobiome in increasing available biomass for the bioeconomy. This could help remove that bottleneck we talked about at the beginning with the plants that went bankrupt in the Corn Belt, help make the bioeconomy more climate change resilient as well, because there will be more stress episodes with climate change. And there's a large potential with regard to further development, current technologies, and development of others, which I'm hoping to be looking for soon. Progress to date, this is the potato field trials. So consortium applied at seeding or at hilling later on, or both times. And nutritional quality assessments again, and greatest increases were for treatments with the consortium was applied in the most pedestrian manner at seeding one time, not at the recommended level. So inoculation increased the in-season biomass by about 27. Well, it was creepy. It's 28, almost 30%, and tuber yield by about 20%. So pretty nice. This is what the data looks like. This is the corn data, and this is the recommended seeding date. This is the late seeding, and here you have the three soil types: clay, loam, and this is with the treatments present. This is one times. This is double the recommended rate. But you can see these guys are causing increases. If you compare this to this guy to this guy, etc. But if you compare this to this, there's nothing much happening. So doing it at twice the rate doesn't really seem to help. So that's grain yield in 2019. This was biomass in 2019. Just to get the student to send me something, and that's what he sent me. And this is potato yield. And again, this is 2018 and 2019, and you can see again increases with the one times, but nothing extra with the two times. And again, the biomass followed the same pattern. So relevance to the industry, evaluated the consortium, the evaluated consortium shows clear benefits to production of both biomass, this is the crop residues, and food. Strongly enhances the market for this consortium. This makes these things of value and or demonstrates their value and promotes the use of biologicals in general, which it's a relatively sustainable approach. So research on novel plant growth promoting strains, clear benefits, and they have been patented. This is the one from the company initially. That consortium was already patented, but the new ones we isolated have been patented now. Supernate Materials is being evaluated for commercial context. It's undergoing scale up right now, actually, and the company is developing a product based on it. So PGPR and their products enhance biomass production in a sustainable way, and so strengthens the underpinning of the Canadian bioeconomy. That's what we want. So just looking here, some of the things to consider going forward: success factors, technical, can new strains be developed? And I would argue quite a lot of them could be. Market, essentially all crop production, but particularly where there's likely to be more. And the business part, growth and development of companies. And we're actually working with 2 startup companies based around this now, one in the context of Biomass Canada and one not. Potential challenges, technical, screening for strains and signal compounds they produce. And we actually, we've gotten quite good at that. So I think we're okay there. Scale up. Scale up is where we kind of start leaving it to the company. And it's all, I've been through this a few times. It's always bumpy and there are always surprises. But anyway, and then technical development demand. So we have to make the industry aware of the potential for the stuff so they want it and want to use it. And this is just in the bigger picture. Success of the project will demonstrate the economic and agronomic viability of the phytomicrobiome members in developing sustainable and climate change resilient production. So that's good. And provide new revenue streams for underutilized marginal lands and crop residues. So some extra income for producers. Future work, we should continue to isolate and evaluate new strains and compounds, and the compounds they produce, which are a type of biostimulant, and combine them with other biostimulants. We should collect further data on the strains we already have under investigation and the signal compounds they produce. We're trying to isolate the signal compound from the Devosia right now. Collect additional data regarding PGPR and biostimulant effects on biomass crops in conjunction with flavonoids to a greater degree. And finally, collect data on existing microbial materials. We just could continue what we're doing for another year. Last slide, summary. So this is an established, what we can draw from this, an established consortium is showing promise outside the greenhouse. Yeah, I guess I should have said this at the very beginning. This strain was originally developed for use under greenhouse conditions, specifically in the production of cannabis. and they came to us and wanted to know if it had field application. It does, and they're really happy with that, and they're selling it much more broadly now. New strains are isolated and are showing substantial promise. These are the new strains we have. Evaluation of other biostimulants is ongoing. We are fiddling with these things in the background. And overall, the approach shows the potential to enhance production of biomass from both dedicated biomass crops and from food crops, and maybe a little more from food crops, actually, facilitating establishment and growth of the Canadian bioeconomy. Yay. Any questions? I see I'm just on 30 minutes.
Yeah, good job, Don. Thanks a lot.
Okay.
So again, that was an update on activity 8. As I mentioned, that's mainly focusing on supporting the production of biomass in Canada. So I just want to mention that that's one of the activity of the Biomass Canada cluster. And thanks, Don, for the presentation. So we have question, Don. The first question is about the stress condition in Canada. So the one question is that which is more critical in Canada, drought or salt?
Okay, so in Canada, I mean, salt. Well, first things first, I should just apologize for dumping a huge amount of material on everybody. That was anyway, but yeah, okay. In Canada, salt is not very often a real problem, right? So we have most of Canada. We have meaningful amounts of snowfall. It all melts, and then we get the soils get a little bit of flushing every year. So it's not too bad in most places. There are parts of the prairies where they don't get enough snow to completely clean up the situation, and salinity is developing in some areas. Parts of the world, about 20% of the world's best croplands are developing serious salt problems. So globally, it's a serious problem, but in Canada, not so much. But as I say, we start with salt because it's easy to apply uniformly. And the other thing is that there's the stress response mechanisms of plants overlap each other quite a bit. So if something works with salt stress, it's probably going to work with drought stress, low temperature stress, heat stress. And we have actually, once we've done all the salt stress work, we often go through it and eventually confirm it with other stresses. And usually if it works with salt, it works with the other ones. So, but yeah, we only use salt stress initially because it's easy, but it's not a huge problem in Canada, to be fair. Yep.
Okay, thanks. And the second question is mainly about, Don, it's about CO2 sequestration. So we have a question that in the perennial biomass crops like switchgrass and miscanthus, what non-destructive methods are used to estimate the reduction in greenhouse gas emission or like CO2 sequestration, especially by root biomass?
Yeah, and we aren't doing it right now. And this is fine in the current iteration of Biomass Canada, but when we apply for renewal for the next 5 years, it will be absolutely critical to have this involve the AFC has shifted now. They want a big focus on greenhouse gas emissions reductions. So we are going to have to put that in place for the next 5 years. And I guess what we'll do is we'll do soil sampling for organic matter contents and things like this, and we'll estimate, we'll know how much biomass is produced above ground and things like this. So we'll get estimates of that, and what the biomass could be used for, the above ground biomass. And we'll run it through, I guess, life cycle analysis.
Mm-hmm. Okay.
But
so,
I think that's what it'll take, yeah.
So do you have any like a specific method you are using?
No, we're not. We're just not doing it right now. And there was no pressure to do it when we started doing this, but there is now. So we are going to have to address this in the renewal application in a very clear way. Yeah.
Okay, great, thanks.
Yeah. Right.
And okay, just one more question came up. Just give me a second. So the next question again is coming from Shahab. How do you create soil conditions that represent unproductive soils?
Oh, there you're back. You moved again. I missed the question. Go ahead.
Yeah, it's how do you create soil conditions that represent unproductive soils?
Yeah. So we're doing what we've been doing is we've been doing it on a range of soil types. So some are better than others. There's no question. And so, but some of them are either too heavy or too light, so they're less productive in that sense. And we've been planting when we do field work. We plant usually at the recommended time and late. If you're planting late, you're much more likely to hit water stress issues and things like this. So we're trying to set it up so we hit some of the kinds of stress conditions we would if we were working on marginal soils. And that's what we do. It's not perfect, but it we have had some clear stress events, and you can see effects more clearly when they occurred. So it's a, but yes, at some point we need to actually carry this to you know, soils that are defined as marginal.
Okay, great. Before we go to the next presentation, I have one question for you. So Don, as you know, the demand for soybean and also canola is growing, especially for the production of renewable diesel, biodiesel, and recently for sustainable aviation fuels. Do you see we have still room to increase the production of canola and soybean on a land basis? I'm not talking about improving the yield, but do we really have more land in Canada to increase the production of canola and soybean?
And you're thinking about this for things like biodiesel?
Yeah, like biodiesel, and as you know, recently for even sustainable aviation fuel, because it's
it's a worry for me, that actually, because when you go back to that, when you're using seed oils for fuel production, you have gone back to the food versus fuel thing, or at least you're really close to it, depending on how. I mean, I have seen people doing it with canola, for instance, with seeds that don't make grade, right? So they would normally be thrown away. That's okay. But the amount of that you get varies wildly from year to year, depending on the growth conditions. So that's a problem. So yeah, it's I guess what I would like to see in the long run is something coming out of pyrolysis or something like this that produces an oil that is suitable for aviation fuel. And that's gonna, there's some work still to be done there. It's I mean, I think some of the pyrolysis oil now has been approved for home heating, but that's a long way from aviation fuel.
Okay.
Yeah. So anyway, all right. It's not a perfect answer, but there you go. So
yeah, thank you very much, Don. Again, appreciate your time and great presentation. And again, just to the participant, you can have access to the slide after the webinar. So again, I'm sure you can have access to the Don in case if you have any further questions.
Yeah. If you got a question, feel free to email me or whatever. You know, it's just, I'm happy to. I'm always happy to chat about this stuff. In fact, maybe a little too much, so be careful.
Thanks a lot, Don. Thank you.
Okay.
So I guess we can move on to the next presentation. That's going to be the update on activity 5, as I mentioned before. And this activity is led by Dr. Animesh Dutta from the University of Guelph. The floor is yours, Animesh.
Thank you, Mahmoud. Let me share our slide. Okay. Hello, everyone. Good afternoon, and welcome to the second presentation.
Sorry, Animesh. So we see two screens. Can you make it just a?
Oh, really? Okay.
Yeah.
Okay. Let me share it. Let's see again. Can you see one screen?
Awesome. Great. Thank you.
Okay. Thank you very much. Welcome to the presentation. We'll be talking today on upcycling of bio-waste for energy, chemicals, and materials based on the circular economy approach. And I am also, along with my colleague, Dr. Pradhan, will be joining as during the course of presentation. So in our presentation, we have 4 sets. One will talk about overall project that we are assigned to and we have been working on. And the second one will talk about some of the research that we have done based on the biocarbon pathway. And another section will be talking about biochemical and biorefinery pathway. And at the very last, we'll talk about technical development, accomplishment, plus future research. So start with our first topics. Let's talk about it here. Pointer here. Thank you. So again, we'd like to acknowledge the generous contribution of Agriculture Partnership, so AFC, and a BiofuelNet, and my partners, Cinder, Custom Steam, Shrimp Canada, and IGPC Ethanol for doing this project. So the overall mandate that we have for this project is to develop a variety of renewable fuel, especially biocarbon, that potentially can replace the fossil coal, and renewable chemicals that can be fossil fuel based chemicals, and biomethane that can lead to natural gas, as well as biofertilizer, especially from non-food and sustainable agri-food waste resources. So here we are very much focused on food versus fuel issue, as Don mentioned already about it, and some of the questions that also arise from there. Soybean and canola, that's what discussed. So our focus is on non-feed, non-food, and which is a lignin-cellulosic based as well as agricultural waste-based feedstock. In order to accomplish those, so our project meeting the Biomass Canada's cluster goal in 3 facets. One is how valorizing this agriculture and food waste, especially we focus here on crop residue, greenhouse residue, and food processing waste. And we developing green processes and define value chain on this. And very last, strengthening the sustainability and bioeconomy of Canadian agriculture and agri-food sector. So the overall project objectives that we talk about, we say that in one of the main theme, waste, actually, there is no what called waste. We consider research just waiting for an opportunity. So it means any waste that we generate today is just not a waste to dumping off or for the landfill. It has some resource, and our goal is to harness that resource and for especially for value-added product. So in that context, when we talk about agriculture and agri-food waste, and we say that agriculture agri-food waste, this is a plant-based, which is renewable, and there is an agriculture farm all around the country, so they are abundantly available in various locations, and they are also considered as a carbon neutral or lesser pollutant. However, when we talk about this to be used as a resource, especially not as a, we found that this has some barriers. One of the barriers is they have a low energy content, they are low bulk density, difficult to like any kind of grass. And when you are talking about the food waste or food processing waste, this is a high moisture content, and they are fibrous and very difficult to make it a small pieces if you want to burn it. And they often come with a high alkali and ash material and alkali matter. So when we talk about this, definitely, so there is some lot of negative. So they are not considered about an ideal replacement for any kind of fossil fuel replacement or any kind of materials that we currently heavily depends on fossil resources. So look into a different technology, and we found that a technology, we call it hydrothermal carbonization, which rename as HTC, biomass. And the way we do it, we treat it in a hot compressed water in a state of drying. So it is something like over the millions of years, the way coal, oil, and gas are produced under the pressure and temperature underground of the earth surfaces, and we are trying to mimic that scenario, especially in the lab scale or industrial scale. And that is what we do through hydrothermal processing. And it gives a lot of benefits because it's cooked like as a pressure cooker. So drying is not required for removing any kind of moisture from the wet biomass. So it doesn't need that kind of energy to process it like other pyrolysis or torrefaction or drying. And this product shows that that low quality agri-food residue can be a potential newer value chain. So the research question in the preview last through this project we investigated is, can we produce industrial grade biocarbon? So that's what our focus, not only the biocarbon, but from this low quality biomass that has a high alkali metal and that should have a low alkali metal and higher heating value as well as higher grindability, mainly for both energy as well as high value material application. And the next one, will there be any industrial grade biochemical as a co-product from this hydrothermal carbonation process water or any kind of revenue that we can generate from this and other waste resources? So in order to do so, we create a number of hypotheses. So our approach in the hypothesis start with changing the operating parameters or hydrothermal carbonation process, and which is mainly the time, temperature, feedstock, what types of we're doing, sizing, etc. And we try to combine and develop recipes so that that can tailor-made to a carbon that we are interested in with a required morphology property for various applications. And in doing so, hypothesis that we should be able to eliminate some of those key barriers that we mentioned for the agri-food waste. And next, once we develop it, we try to develop a little bit higher value chain. So we propose two-step thermocatalytical process, and we hypothesize that will be able to generate high-value carbon materials. And also, we also hypothesize that hydrothermal carbonation or HTC process in combination with anaerobic digesting system and those from the agriculture, agri-food farmers and working in that area, we understand that when we talk about anaerobic digestion and the steps that rate is considered as the rate limiting steps is the hydrolysis. HTC carbon, we also produce HTC process water, which is a kind of acidic. So we hypothesize that when we integrate this HTC process water with anaerobic digestion, the hydrolysis process will be enhanced, and that leading to have a higher gas yield and so on. To approach, establish this hypothesis through a number one. First, we develop with the hydrothermal carbonation design and development of the process. Then we try to characterize those feedstock that will be used as well as the product that we generate. And we try to develop an industrial or a process that can lead to a commercial processing technology. And our critical delivery would be based on this. We try to use life cycle analysis as well as life cycle cost analysis. So if I want to say that the technical accomplishment, there are 6 objectives of our project. Starting with the first objective was to pre-process all the greenhouse residues, energy crops, and even municipal waste bin or the food waste. And we already reported that in last couple of webinar, and I will also highlight a little bit in this section as well. And there are some work that we have accomplished during this 2021-22 years is characterized and of the hydrochar we produce as well as process water. We have done a number of work and now work on the effectiveness of the hydrochar, especially in the activated carbon and preparation, as well as doing some work on water detention experiment, nutrient recovery experiment from the wastewater. And we have completed this study. Now data is in under progress. We also use the effectiveness of this anaerobic digestion, process water in anaerobic digestion, and we show that this will enhance the biogas production, and which already completed. And we also see the potential of using biogas and hydrochar in a combined heat and power together as a closed loop based circular economy. And we have performed this work, and we have performed a numerical study on that. And the very last work that is pending that will be done in 2022-23 is mainly the environmental and economic footprint based on LCA and LCC analysis. So just to recapture, we have done a number of studies on biocarbon development research, and that biocarbon development research, as can be seen, we start with waste, woody waste, purpose-grown feedstock, green. We have done more than that, like grapes pomazes, apple pomazes, cranberry pomazes. We haven't shown it here, but all kind of things to produce a hydrothermal or HTC carbon that at the first steps here. And then that can be again upgraded to the biocarbon for the soil amendment. We have done work already on the to see the nutrient recovery on this. And we have also done biocarbon for the heat and power production. And we have done some work on the or high value PCI pulverized coal for steel and iron industries. And our future direction, we are positioning towards activated carbon for energy storage and wastewater treatment. So we demonstrate that when we produce the hydrothermal carbon or HTC carbon, that exhibits towards the coal that used for the power plant or power generation, and that's able to reduce the some of the unwanted mineral, something like potassium, sodium, calcium within the ash matrix itself. So that limits the you reduce the erosion and corrosion due to when these are present in the fuel. So that's barrier we are able to demonstrate it, and we are able to show it through hydrothermal carbonization process. So we can see that remove those kind of barriers, and depending upon application, it may require further processing, as we can see from all those. So this research has been moved to upper version, where we are able to produce a number of publications as reported here in the last 2021-22, and especially we develop a mathematical model from the HTC carbon to understand different processes, and we are able to develop a continuous HTC reactor. I will show it later on, but based on, and these are published in energy and different journals, as well as we are able to integrate HTC with anaerobic digestion to produce power, and we compare with the base scenarios. We are able to produce biocarbon, biomethane, and biofertilizer from plant residues. That's published in Energy Journal. We also are able to separate CO2 from the biogas, especially to going towards renewable natural gas. And we have also done considerable amount of work, natural gas from the organic waste and opportunities for Canadian pipelines. And going forward, we are able to upgrade this biocarbon even to produce some of those biochar for hydrogen storage material, and shows a kind of potential. And we develop biocatalyst based on this carbon, and that's we'll be able to, we hope that able to produce direct biodiesel from the lignocellulosic biomass, because that's shown